the cytoskeleton of the cell, biosignaling, which is basically like cells texting each other in a way, and the mechanisms of actual cell development. So continually when we come and think about what is life, what are cells made out of, what are we kind of looking at, our four building blocks are continually going to come back to amino acids, which are going to help us build proteins, carbohydrates, which are going to obviously be part of sugar. They're going to give us energy, there's storage. We're going to see other functions of carbohydrates today. Lipids, which we know is like fats and oils, they're obviously storing energy as well, but they also have other functions, which we're going to see today. And then nucleic acids, which are going to be that genetic information. So it's important, I would say, when you're thinking about these different building blocks, is that you recognize like a picture of them. So if you see like a base standard of like an amino acid with the amine group and the carboxylic acid and the functional groups, you understand like, oh, that's an amino acid, that's a protein. Hopefully you're in the point where you can actually see one of those and say, oh, that's, threonine, or that's serine, whatever it is, and really just getting really comfortable with those. So you want to be able to recognize, like, if you see a drawing that you know exactly what it is, you can label it as one of these four categories, but you can also kind of understand maybe what bonds it's making, maybe what function it might have in the cell, just kind of like some other information about it. Oh, I wrote down here, Simran was the person who I guess taught the lecture about this in carbohydrates and biomolecules, on like mainly carbohydrates probably at that point, but also about different biomolecules. So you can definitely go back and review that lecture. If there's anything that seems kind of uncertain, definitely feel free to throw it in the chat so that we can look over that too. I think the biggest message that I definitely put emphasis on last week, but is that structure determines function. So the structure of, we're going to talk about a lot about the plasma membrane today. The structure of the plasma membrane determines its function. The structure of different like signal proteins determine the function of the signal protein. And so all of these things, like, we can continually coming back to either thinking about what is the structure of this molecule, cell, organelle, and what is its function, and how can I understand those? Anytime you see a question that's asking you about anything cell related, you should be thinking about structure and function. So this is really important. You're going to see me repeat it over and over again, pretty much on all of the slides. I've got the function labeled right at the top, and I think anytime you see that, you can think about like, what are kind of like functions and how does the structure help that happen? So we're going to start at lipids. Particularly, we're kind of diving towards lipids in the membrane form. We know that lipids obviously have other functions that they are a part of, but today we're kind of focusing on the lipids to membrane journey. And so when we think about that, often you will see lipids kind of or fat molecules kind of depicted like any of these. So when you see a structure that has this long kind of tail thing and a carboxylic acid group on the end, you should immediately be thinking lipid. There are different types of lipid molecules. So we've got here what says fully saturated fatty acid, trans unsaturated fatty acid, or a cis unsaturated fatty acid. You definitely will hear about these things. Like in nutrition, we talk about fatty acids or the unsaturated or trans fats being like, potentially worse for you. And so when we look at this, we have to remember that off each of these are each carbons, right? Which are going to make up this hydrocarbon tail. But the other part, I guess I just gave it away, is that attached to each of these are hydrogens. And so when we're thinking about the difference between something that's saturated and unsaturated, it has to do with the hydrogens. Hydrogens is like in terms of saturation. And so here we see we have a double bond, and we know that we can only have, right? Carbon only makes 4 bonds. So when we have a double bond, we have C double bonded to C, and then they're all part of, the rest of its chain. So this actually already has 3 bonds. So now it only has 1 hydrogen here and 1 hydrogen here, compared to other kind of carbons that are going to be in this chain that are going to have 2 hydrogens each. So that's really the difference in the naming convention between a saturated fatty acid and an unsaturated fatty acid. Now, probably like you're familiar with from chemistry, Trans and cis have to do with the orientation around the double bond. So we can see here that in the trans double bond, basically our two adjacent carbons are going to be in the different plane compared to a cis double bond. They're on the same. And you think about like the planes, I'm really not, chemistry is like not my thing, but these are on different ends of the plane or different, one's going up, one's going down compared to both being on the same side of the plane. And you're also thinking about it in relation to, the hydrogens that are there. So you can see here, if I drew them in, that's the bad color. The hydrogen is up here compared to probably the hydrogen is up here, just thinking about displacement and order. And then compared to having hydrogens kind of on this side. So those are the differences. Now, when we think about, the structure of these, it's going to determine part of their function. So part of what I've included here is that the differences in what state they're in at room temperature, compared to also the differences in their melting point. If we think about all of these hydrogens that are going to be, in a saturated fatty acid that are going to be there, that's a lot more bonds to break. And yes, well, you could argue that a double bond is going to be like more to break. Oftentimes we see those repeating. And so this is going to have a higher melting point. I have to put more work in to actually get all of this saturated fatty acid to fall apart and basically come down to a melting point. So at room temperature, saturated fatty acids are often solid. Think of like butter, think of bacon grease. Those are going to be those are partly made up of other stuff as well, but saturated fatty acids. Compared to when I think about an unsaturated fatty acid, that is going to be liquid at room temperature. Think about olive oil, think about canola oil. Obviously, we can, take these naturally occurring lipid molecules and modify them in a lab so that they will, potentially appear healthier. There's a lot of belief that, olive oil and avocado oil are potentially healthier for you than like canola oil. Really, none of them are like wildly healthy. Hate to break it to you, but you should use them in moderation. But you can see kind of differences between like a butter form of like, technically that's still also like an oil. Yes, it has milk solids in it, but even when you like melt bacon grease, right? If you're a vegetarian, I'm now talking about like meat products, but if you made your bacon, you have the fat that kind of is released from the protein as it's cooking. It then looks, really glossy and shiny, and maybe you like to like dip bread in it or something like that. But if you leave that pan on the counter, on the stove, and there's no heat being applied to it, after time, you'll see that will become solid. And that's because most of the lipid molecules in that bacon grease are actually going to be unsaturated. So they're going to be solid at room temperature. Now, the reason that this is important for us, and I'm sorry if this like feels like review, is because this is going to potentially change the way we think about lipid molecules that are going to become part of the plasma membrane. When we are going to think about how temperature affects something called fluidity. And so we need to kind of remember these basic basic points. So the bacon grease, is solid at room temperature. So it's going to be saturated. I'm sorry if I said the other. It's going to be saturated. So I can put up here like bacon grease and butter. Think about those kinds of things. Butter compared to like olive oil. Very different kind of states at room temperature. If you, sometimes this does pop up on the MCAT, so I just wanted to include it, but fatty acid like nomenclature is kind of going to follow a procedure like this, where you have different kind of like, I guess like numbers. It's different than like nomenclature of like when you're naming like chemical or like, kind of like compounds and being like, oh, that's a 2,3 carbonyl, whatever it is. Penta something acid. I was terrible at that. But in fatty acids, it feels like it's a little bit more straightforward because basically the system is always going to be our carbonyl carbon at the end here. This guy becomes our carbonyl carbon because he's attached to the carboxylic or the carbonyl group. And then the rest is going to be kind of our fatty acid chain. So we're going to start our numbering. This then becomes number one, the carbonyl carbon is always going to be number one, and you're going to count the tail. So sometimes I think in one of my MCAT, maybe it was a practice, can't remember if it was the real thing, they asked you to like actually like what's the fatty acid nomenclature for this molecule? And it was like in front of you written like this. So it's just a good skill to have. So you're going to count from the carbonyl carbon to your tail. This is going to be basically the number of carbons in your whole chain, compared to here, this 5 number is going to be the number of double bonds. I'm going to signify that as here, double bonds. And then you've got your little delta, and this is showing me where my double bonds are. So that's going to be just like a really quick kind of like simple way to describe it. Sometimes you'll be given just the nomenclature and be asked to choose which image best depicts that. So you can be asked either given kind of like given the drawing and asked to produce the nomenclature, or given the nomenclature and choose the best drawing that matches that. So it's just something to be familiar with. You may have seen it in practice questions. So I just kind of wanted to throw it in here. And then we've also kind of got this other part of the nomenclature, which is going to show us the position of the last double bond from the end of the chain. So my end of the chain here is 20. And if I go back 1 carbon, 2 carbons, 3 carbons, this is, oh, that's like a terrible. Basically, if you go back, this is going to be 1, 2, 3. That's the way it's going to be described. So let me just do that again. This is showing me where my last, I'm just going to put that for double bond, is located from the end. That's very important. So if this is my end, 1, 2, 3, it's 3 carbons from the end. And sometimes they'll give you this information also to kind of like also help you pick if they gave you just like the nomenclature information and asked you A, B, C, or D, which of these is the accurate representation of the nomenclature, you'd be able to match it. And this information can be really helpful sometimes when deciding between 2 different kind of like depictions. Sometimes they include it, sometimes they don't. Sometimes they never cover this, but thought it was worthwhile to put in, and maybe you've seen this before, maybe you haven't. Any questions kind of so far in just like fatty acids general? All right, we're going to keep going. So we know that fatty acids, that we don't always just see them in those one individual chains. So sometimes they can undergo dehydration syntheses with different kind of molecules to produce other different molecules. So one of them we hear about a lot is triglycerides. You might have heard of a phospholipid or a sphingolipid, different kind of things, basically different attachments onto a lipid. And they can attach typically in the kind of like what we're going to consider like the head position, and these hydrocarbons, which is just a long chain of carbons with hydrogens attached, become what we consider like the tails. I've just got a quick question is here. No, we do not see alpha and beta necessarily mentioned with those compounds in naming conventions. So when we're looking at different potential options for I guess like more advanced maybe lipid molecules, in a way kind of like you have like a string of amino acids becomes a peptide chain, and then different peptides joining together might be a polypeptide. This is kind of like a polylipid in this case. So we have 3 different fatty acid chains here. And you can see kind of like their little carbonyl group at the end here. And that is going to undergo a dehydration reaction. So we're going to spit out H2O, in this case, 3 H2Os, in order to connect with glycerol. And that produces a glycerolipid is sometimes what it's called, but most often a triglyceride. And if you think about that, tri is 3, right? And it's a glycerol, so it's going to be glyc, and then ide is something, glyc. Ides are sometimes seen as lipid molecules. So that's going to be how like how that's going to produce. And then in this case, it produced 3 different water molecules. So we can have fatty acid chains basically join with a lot of different options. Sorry if that feels, that was a weird transition. So we mentioned glycerol. You can also have something called sphingosine, which is another different molecule. It's an amino alcohol, also has a really long carbon chain. So this part right here is the actual sphingosine carbon chain. And then you can see here that we can also have phosphate groups also join in. And the benefit of kind of having these like attachments is that it changes the behavior of the actual lipid molecule. So if we go back, or if we had our original kind of lipid molecule, which I'll just quickly draw one, a very messy one. Other than here where we have some negative charge, most of this is relatively neutral. Carbon's relatively neutral. Yeah, hydrogen's like a little bit positive, but there's not really much going on. This is a very like amphipathic molecule, very neutral molecule, kind of under the radar, is going to probably act hydrophobic in water because it's got all of these like neutral molecules. So the benefit of potentially adding on a glycerol or adding on a sphingosine or adding on a phosphate is that we can then change the electrochemistry of that molecule, and therefore we can have different interactions and different kind of connections and different basically behavior. And so you can see over here, it's kind of broken down in this tree. We've got storage lipids, going to be hydrophobic. We've got our triglycerols, triacyl glycerols. There you go. And we know that they have a ton of bonds, and so therefore they can store energy. Maybe you don't like the way that this is storing energy in your body, but it's true. It's going to happen. It's important for us as well. Definitely has a lot of functions. So that's kind of what we think of as like maybe like fats and lipids in like a physiological, or like if we're thinking about like maybe kind of in like more of like the, I'm not really sure what system it would be in, but like thermoregulation. As animals, we sometimes need to have, we need to be able to thermoregulate, and our body can do that by burning fat, which is a really great way to get energy. Or potentially if we're overeating, we're going to store that excess energy as fat. And so this is kind of like this storage kind of like mechanism compared to when we have membrane lipids, which we're going to see as amphipathic molecules, which are going to have differences in their polarity. And so I think earlier I said that, it originally was amphipathic, and that was incorrect. Sorry, that like phospholipids and sphingolipids are going to be amphipathic. So they're going to part like water and part repel water. And so that's kind of their duality of nature compared to being hydrophobic, which is what our storage lipids are going to be. So sorry for that kind of confusion there. So when we're thinking about interacting with water and kind of the behavior of lipid molecules, it's going to be important to us that they have this part that likes water and that's going to interact, and that's going to be the more polar end compared to the like hydrocarbon tails, which are going to necessarily be like hydrophobic and afraid of water, and so when we think about here, looking at phospholipids, this is what we're mostly going to see in the lipid, in the plasma membrane, which is sometimes called a lipid membrane, a bilayer membrane. It's called the fluid mosaic model. There's like a ton of different names that we can think of to talk about the whole plasma membrane. But when we look at actually what's making up the plasma membrane, it's most likely going to be these phospholipid molecules. So you can see here, they've got a phosphate group. They've got a ton of oxygens, one that even carries a negative charge. There's a ton of resonance in here, which is why it's happy to be in this little state. But basically we have this really kind of like, that looks like a double bond, this really negative kind of like top end to it. And then these tails, which are just very happy carbons and hydrogens, and they're going to be technically our more positive end due to polarity, but really they're not charged. They're just going to be happily hanging out there as long as they're not in touch with anything that's polar. We know that most liquids, obviously you can find a nonpolar liquid, but in our bodies, cytoplasm really is similar to water, and that's going to be polar. So it's going to be important for us to kind of remember that. though we've got kind of this duality in the nature going on because of the polarity of the compound. So if we just took a bunch of phospholipids and we spontaneously put them in a liquid environment, we're going to see them automatically form mostly probably the first two if it's just a little bit of liquid, but they are going to want to organize in a fashion that's going to kind of like basically protect the hydrocarbon tails from exposure and expose the phosphate heads to the polar like molecule that they're interested in kind of interacting with. So you have here what's called a liposome, which usually what happens when there's actually liquid inside here, which this picture is kind of like hard to depict that on, but imagine there's also liquid inside of here. And that's important for a liposome, that there is liquid inside as well as like liquid surrounding out here. And that can be really important when we're thinking about the way that like even like medicine is delivered into our body. Sometimes we coat basically the outside in oil to protect it. And we're trying to protect this liquid that's inside so that it can pass through our digestive system and kind of slowly be released into our system. And it's not just immediately dissolving. There are other forms of medication where we do want it to be immediately dissolving. So maybe if you're interested in like pharmacology or potentially biomedical kind of design or biopharmaceutical design, this is something that's really interesting kind of in that phenomenon, is the fact that lipids naturally do this. And so we can actually use this to potentially protect liquids that we're trying to transport to other places of the body or have a slower release into the bloodstream. Compared to a micelle, which is just a droplet of oil completely, there's no water inside of that micelle. If you have a cup of water on your desk and you added any form of oil, olive oil, you would see those little beads of oil. We can see them form. There's no water inside of there. And so this happens in the body physiologically during digestion. When you eat something, I just had like a wrap for lunch. There was like some yogurt in there. There's going to be some fat particles that are going to be digested. When that's going through my digestive system, you may have seen this term micelle come up, which basically are like clusters of these lipid molecules that are all going to hang out together. Eventually they will be broken down, and that's what we use our bile for. That's what different acidities in our stomach are for. And so we've got a lot of ways to kind of break that down. And then lipases, of course, are going to get in there and hack it apart. The third kind of spontaneous arrangement is in this bilayer sheet, where we can see that we have these phosphate heads on the top with the hydrocarbon tails facing each other, and then a second layer doing the opposite. And this allows us to have liquid both on the outside and on the inside, which kind of leads us to like our big reveal that this is what's going to happen in the plasma membrane. Exactly. It's a hydrophobic effect. It's naturally occurring. So this is something that's really, really important for us, is that these are naturally occurring patterns that are going to spontaneously happen. We're not having to like manufacture this. You know, the fact that we can be humans and be kind of functioning is basically based on this. So you've probably seen many pictures and many drawings, many depictions of the plasma membrane. And there's going to be like some other stuff in there. You know, we're going to go over kind of what these different things are. But because of this ability to form this bilayer, where we've got liquid on the outside in the aqueous environment, and then our inside liquid, which is going to be our cytoplasm, we have this bilayered effect. It's important to remember that these are actually different than liposomes, because while liposomes do have water in the center, it's usually just like a drop of water. That's all. There's nothing else in them. They don't have any proteins inserted in the membrane. You could think of it maybe as a giant liposome, but it's important to remember like a cell is going to have transmembrane proteins and recognition proteins, and it's going to be doing like a whole other stuff. It has a nucleus. It's got a lot more going on. But if it helps you remember that like liposome is closer to cell than a micelle is, that's totally fine. So I did mention earlier, it was called this like fluid mosaic model, and fluidity is going to be important as we think about signaling cascades and different channels or transporters that are going to be kind of in the cell. And it's also going to be part of the procedure of like cell death, programmatic cell death. So we're going to keep going. We've got 2 review questions here. They're just true false questions. And you can just write in the chat kind of what you think. If I find a cup of unknown oil at room temperature, it's liquid. Oh, and then the question is true or false? There are few double bonds in this lipid structure. Just generally, don't get too detailed. Remember that we found it. It's liquid at room temperature. Is it going to be saturated or unsaturated? Yeah, it's going to be unsaturated because we know that unsaturated fatty acids are going to be liquid at room temperature. So we're looking for unsaturated. And if we know that it's unsaturated, that means there's presence of a few bonds, and there's most likely going to be a lot of double bonds. Yeah, so the answer here is false. Great. Second question, true or false, all lipids are hydrophobic. Oh, sorry for the doorbell. And my dog, sorry, she's gonna bark. Yeah, it's false. We know that we have, sorry about her, phospholipids and sphingolipids, which are gonna add to the polarity. So those are gonna be what we call amphipathic, which are gonna have a hydrophobic end and a hydrophilic end. If this question said like all kind of like fatty acid tails are hydrophobic, that would potentially be true, but this original one is false. Okay, we're going to move on. How are we doing? Feel free to like give a thumbs up. Feel free to chat any questions in before we kind of move on. We're going to talk about like membrane fluidity, parts of the plasma membrane, all the proteins, all those fun stuff. Thanks, Jonathan. Thumbs up. Pace is better today, hopefully. Excellent. All right, we're going to move on. Great. To membrane fluidity. So we talked about this like idea of like a fluid mosaic model. We don't want necessarily, right? We know that most of our cells are going to be like this circular item. So we already need to have like a little bit of flexibility going on. We're not looking for something that's super going to be rigid and tough and kind of like difficult. So in order to have this flexibility, we call it in regards to the plasma membrane fluidity. So that's going to be kind of like an important term. Whenever you see that fluidity and you're in the bio section, you're going to be thinking about the plasma membrane and the ability for it to like move and be flexible. Obviously, it still needs to like hold its circular shape, or potentially maybe it's more of an oval or whatever the cell's doing at that time, but it needs to be able to adjust. So some key factors that are going to impact our fluidity are going to be the number of double bonds. Basically, the more double bonds you have, the more fluid it's going to be. And this should make sense to us if we're thinking about, remember our hydrocarbon tails, if we've got all these saturated hydrocarbon tails, they're going to sit really nicely together, right? But if we've got, compared, I'll draw it in a different color over here, potentially some double bonds, those aren't gonna, this is a double bond, those aren't gonna sit together as well. So this is also something that contributes to the fact that unsaturated fatty acids are going to be liquid at room temperature, is they aren't able to compact next to each other as easily. So when we're thinking about it, I'm going to scroll back up to the other picture really quickly. When we're thinking about it kind of in our plasma membrane here, in our bilayer, if we think about these little tail guys sitting all next to each other, they can sit all nice and perfect, saturated tail after saturated tail after saturated tail after saturated tail. But if we need a little bit more flexibility and a little bit more fluidity, which is what we're calling it, then it's actually going to be beneficial to have unsaturated fatty acids in there to kind of like throw off the pattern a little bit and create some space. And this is going to be particularly helpful when we have these other kind of friends or players in the game of different proteins and kind of different things going on. So it's important to remember that double bonds, basically unsaturated fatty acids, are going to increase our fluidity, especially, which makes sense, cis double bonds, as I kind of drew here. Ooh, that was my eraser. As I kind of drew here, because they are going to really throw a kink in that pattern. They're going to take up space because of the way that their bond is arranged. And sorry, Michaela, I was just reading your question. The way their bond is arranged. You said, that's why I thought the review question was true, because most lipids with unsaturated bonds have fluid. Yes, that's true. But the question said it only had a few double bonds compared to, like, I guess it was confusing because I should have said no double bonds or double bonds. I was trying to go for like few or many. So sorry if that was confusing. I can see why you thought that now. So trying to more, see the difference between like just a few or like tons. Something that's going to be liquid at room temperature, we have to think about being like really unsaturated, thinking about it having many double bonds, kind of like the example I showed in the nomenclature picture. So we've got double bonds are going to increase our fluidity, especially when we have cis double bonds. At higher temperatures, no matter what is present, both saturated and unsaturated, we're going to have more fluidity. This makes sense. Typically higher temperature, higher energy, more movement, more stuff happening. That should like all jive in your mind. However, the only thing that throws it off is cholesterol. Now we know that cholesterol is a steroid, but it's actually made from lipid molecules. You might have gone over this in the biomolecule stuff. We've got a little bit on cholesterol today, but this is a cholesterol right here. Ooh, that's my highlighter. This is a molecule of cholesterol. You should be pretty confident, like recognizing that. Typically it's 4 rings, but it's 3 6-membered rings and 1 5-member ring. Yep, excellent. It looks like 3 houses, the 6th carbon, and a garage. Nice. So that's a great trick. It should just be one of those things you see that molecule, you're like, cholesterol. It's a lipid, it's a hormone. Boom, we've got this is considered like our little hydrocarbon tail out here to the side. Cholesterol is really popular in the plasma membrane. And this is because cholesterol is like, it's really good for structure and support. And so it wants to be kind of like a party in there. It is mostly hydrophobic. So it's usually kind of like wedged in between, likes to hang out with the tails. But it's going to affect our fluidity. It's basically a member of this, lipid family. So it's going to affect our fluidity. It kind of does the opposite. So at high temperatures, when we're usually really fluid, cholesterol is going to decrease our fluidity. And at low temperatures, when we would usually be kind of more rigid, it's going to increase our fluidity. So this is actually beneficial to have within our plasma membrane because it kind of builds in this balance for us. And so when you're thinking about fluidity, you're always trying to think about what's naturally happening and what's going to kind of bring me back to balance. So naturally at high temperature, super fluid, I might be risking like rupturing my membrane, or potentially it's too loose, there's not enough stability. And so cholesterol kind of brings you back a little bit, or potentially at a low temperature, right, you're going to be more like rigid. Think about things that are kind of freezing. They're tensing up. They're getting a little bit tougher. And so your cholesterol is going to add to the fluidity. You might also throw in some unsaturated fatty acids. Those are going to add to the fluidity so that you can kind of keep this balance of being like in this peak fluidity zone. There's no like measurable quantity to that. It really is cell to cell dependent. If we think about like different cells in our body, obviously like our skin is a lot softer than our nails. That has to do with the fluidity of the cell membrane and potentially products that the cell is producing, the cell type, all of those different things. But we know that the structure of our cell, the fluidity of our cell membrane is going to determine its function. So if it's really soft and really fluid, it's probably going to be maybe like a blood vessel or a blood cell, compared to if it's less fluidity and it's really rigid in its cell membrane, it might be a bone cell. And so those can kind of help determine where it's going to end up, or going to be programmed obviously by the DNA, by the information that is, kind of dictating where this cell is going to go and what it's going to do. So those are this is like the overview of fluidity. Question again here. what would make our body temp change enough at the cellular level to change how cholesterol impacts the fluidity? I can only think of something if I've had a fever. Yeah, so it's not necessarily, I wouldn't think about increasing or decreasing the amount of cholesterol. Just think about where the cholesterol may or may not be. So there may be like in cells, in your bone cells, you're not really going to be storing as much cholesterol in those cells because their like cell membrane basically is just going to be the same fluidity at all times. Whereas like if we think about maybe like our skin, sorry, that's my dog in the background. If you think maybe about our skin, there's definite exposures of lower temperatures and higher temperatures. And so there may need to be more cholesterol in like the skin cells. So think more about like the location of the cell, I guess, exposure to heat compared to like changing the amount of cholesterol in a certain cell or not. Yeah. It's kind of one of those things, but most animal cells have cholesterol in their cell membrane because it also is like a really good kind of like structural support. And it's obviously going to help. It is technically a hormone, so it's going to help in that hormone signaling as well. So another question, hopefully this clarifies from the last one, but which fatty acid tail would contribute most to the stability of a cell membrane of a thermophilic bacterium? These are sometimes like, and a couple of you were asking me about terminology. So like thermophilic, being that thermo is going to be heat and philic means it's loving. So it's going to be something at high temperatures. We'll call this one A, this one B, this one C, and this one D. All right, we've pretty much got one from like each answer, 2 for D. Okay, a few more for D. All right, so let's think about it. So we know that this is heat loving. So we already have, right? High heat means high fluidity. And we're looking for something that is going to stabilize it, which means we're looking in a way to almost decrease the fluidity. So in general, right, when we compare A and B, they have double bonds. C and D don't have double bonds. When we think about kind of double bonds in general or unsaturated fatty acids, those are going to increase the fluidity even more. So we can already say that A and B are not good options because they're helping us increase the fluidity when our goal right now is to decrease the fluidity because we're in a hot environment. And then this is like I think this is straight from I think I got this from Khan Academy maybe, or another MCAT prep, but this is like really similar. MCAT will love to give you two options that their C and D are both correct, right? The only thing that's different is that D is a little bit longer. And so we presume basically that the longer fatty acid chains are going to be able to be more stable. They're going to compact together. And so this is going to be our best answer because it's saturated. So it's going to add to stability. It's less fluid and it's longer. Does that sound good? Monica and Sophie, does that kind of make sense walking through it that way? Totally fine. I think it's really important. Someone asked me in office hours, like what tools I used. I never really used the highlighter kind of like tool, but I think this is a really good question to remind yourself, right? We just spent a long time talking about fluidity, and now the question is asking about stability. So sometimes it's nice to just like when you're reading the question, remind yourself like, what is the goal? You know, like what am I actually looking for? Okay, I'm looking for stability. So I need to do the opposite of kind of what's going on. That might be a case where I would use like the highlighter tool. Okay, yeah, totally fair that a longer one might not be ideal in the membrane. Of course, if you saw other, like compared to the other fatty acid tails and they were all short, being the shorter one is going to be more beneficial. In this case, since we don't know what anything else looks like, we're just going to go for the one that's going to have basically this long kind of like tail. It's going to be adding to our stability, and we're hoping it's just like packed again with all one cell look similar like it. But yeah, that's fair, Monica, to think about the others. But if we don't know about the others, then we're just going to presume it's going to be exactly multiple copies of our answer. Okay. So when we think about, as we've talked about, structure determines function, we've now gone over the structure. We know that we have these phospholipids that are kind of going to be like arranging in our bilayer. So we've got kind of like our outdoor layer, which I tried to use a pencil, but came up with a marker, right? So we've got our outer layer, we've got our inner layer. I'm drawing these like very simplistic, and all of our tails kind of facing in between. We have to think about like, what does this actually do? Well, we obviously know it's going to give the cell, some shape. And so that's going to be a part of structure. We also know that basically this forms a barrier. If we think about kind of like our extracellular environment out here and our cytoplasm inside, the only thing that's really preventing something from coming in is this plasma membrane. So therefore, it's going to function in protection. And this is going to be important for like any kind of like invader cells, or maybe an invasion of a virus. It's going to be coated with lots of fun things that we're going to kind of talk about out here that are going to kind of aid in that. When you think about immune responses and antibodies, a lot of that is happening on the plasma membrane. Yes, it's going to send maybe a signal into the cytoplasm and into the cell to change something. But a lot of that first line of defense is the plasma membrane and what makes it up. It's also going to help us with transport, which we'll see later. We might be moving things out of the cell or into the cell, depending on what's happening. And then also recognition. We're going to go over just different kind of components of recognition, maybe cells having an identity themselves. Like even when we think about blood cells, if you know your blood type, you might be like O positive or O negative. The positive or negative is the presence of a glycoprotein on the outside of your blood cell's membrane. So even things like that can be applied kind of to any kind of like real world application. And then we talked about the fact that most of these are phospholipids. We've definitely got some cholesterol in there too. Also, sometimes in cell membranes, you can see waxes, and these are just another lipid molecule, and they kind of help to make things waterproof. If you've ever kind of like looking in your garden, or maybe you really love to make flower arrangements, you might have noticed that certain plant leaves almost feel like they have like a wax on them. And this can be really protective, particularly if they're in a really hot environment, or there's not a lot of water. Maybe they're kind of being like damaged repeatedly, or they're like a bush on the ground, so they need to have kind of like this extra protection. So sometimes you can see that wax in there as well. That's like another part of like the protection function of the cell membrane. We have it, both plant cells and animal cells can have waxes in their cell membrane. Humans have it most likely when they are thinking about, like, if you think about like your nails, right? You have kind of like a waterproof type thing. Barrier is the word that I wanted there. And so that's because of the waxes that are going to be present in those cell membranes. Obviously, we're also producing like nail as well, but can be also in like the cuticle, particularly. That's where we see like the waxes in our cells. So moving on, we've kind of like set up the big picture. So one of our functions of our cell membrane is transport. So we're going to kind of go now deeper into transport. Transport basically can be broken up into two different categories. Either we've got passive transport or active transport. The biggest kind of differentiation between these two is that passive transport is going to not require any energy, and it's just going down the electrochemical gradient from a high concentration to a low concentration, which is kind of like depicted here. Basically, all of these solutes are in high concentration and they're moving to low concentration if their arrow is going with the concentration gradient, compared to active transport, which is going to need energy in order to move the molecule, which typically means that the molecule is going to be either be moving against its electrochemical gradient, or sometimes it's just really large. So it needs kind of help. So when we think about passive, we have two types, and these are just going to be kind of vocab words that you're going to have to know when you're reading. It's just really going to help you kind of like speed up a little bit. So you've got simple diffusion. These are just going to basically, as it sounds like it, just pass right through that lipid bilayer, compared to facilitated diffusion, which is going to be facilitated almost like it's going to be helped out. Maybe it's going to go through a channel, maybe it's going to go through a transport protein. There's going to be something kind of helping it get through this molecule, get through the membrane. Compared to when we think about active transport, there are also going to be two categories. I would say these are less common terms, but it's good to be familiar with them. You have primary active transport, which is just basically using energy to simply get something from one side to the other. So glucose is a really good example. Takes energy to get glucose into the cell. And sometimes it's going against its electrochemical gradient, sometimes it's not. So potentially it could be in passive or active. If it's passive, it's facilitated. If you're actively trying to get in there, it's going to be primary active transport. But that's probably a confusing example. So actually I'll use a different one. So for primary active transport, it's use of energy to transport a molecule against its gradient. So if we think about a sodium-potassium pump, we're using these pumps to reset the electrochemical gradient for a cell. But however, when we're doing this, we're basically trying to pump them against their gradient. And so therefore we need to use energy to put that in, compared to like a secondary active transport, which is going to be energy from one thing is going to move something else in. And so the one that I always think about this is typically like at the end of cellular respiration, the ATP synthase. It's basically the energy from from using the ATPs pumps the hydrogen against its electrochemical gradient. So that's the one that I usually go to. It's probably unlikely that they would say, tell me a molecule or tell me a protein or an enzyme that's a primary active transporter. Sure, is it possible? Yes, but that's really just like a memorization question. And few questions on the MCAT are truly memorization questions. So I wouldn't be as concerned with kind of being able to name them, but it is nice if you're reading a passage about maybe something moving across the cell membrane, you can start to think, is this passive? Is this active? Is this going to be facilitated? What is actually moving through, the membrane? Is it going with its gradient or against its gradient? So these are kind of some of the things that should be popping up for you. Typically, if you had to like categorize, passive transport is going to be really small molecules, gases, non-polar things, compared to active transport is going to be like anything with a charge, big kind of molecules, which is why like glucose gets in there, proteins, potentially antibodies. We're looking for kind of like bigger things that are going to be brought in. So when we kind of move to this other cell, this is simply talking about what is part of passive transport. So you can see O2, CO2, and N2, those are all going to be gases. And then steroid hormones, they're able to make it through. They don't usually make it through. They often, most of the time, actually hang out within the cell membrane. And that's how they then run their signal transduction cascade. But typically, most of the hormones are non-polar. So they kind of slide through. And when we think about this, cholesterol, which is not a steroid hormone, but is a hormone, is also present. But those are kind of going to be the things going via passive transport, whereas kind of any of these bigger molecules, water, bicarb, glucose, or things that are charged are going to be, they're going to need help. So these guys are going to be part of, I guess this is like passive transport, but also diffusion is what it's kind of showing. And these are either going to be facilitated or active, depending on their gradient. Obviously, the gases also depend on their gradient. If they're moving against their gradient, that's also not going to work. But typically, O2 is higher outside of the cell than inside of the cell. Same for CO2 sometimes, things like that. So when we're thinking about, here's a little question for you about the plasma membrane. Which of the following best describes the composition of the plasma membrane of an animal cell? A, cholesterol esters, proteins, and a small but significant amount of triglycerides. B, phospholipids, sphingolipids, cholesterol, and protein with some carbohydrates. C, lipoproteins, in which triglycerides are a major component. D, a lipid bilayer formed mainly of cholesterol with a protein attached to both sides. Anyone else want to guess? Throw a guess in? Some for B, some for D. All right, so this one is a little bit tricky, but we'll go through it. So the first one says cholesterol esters, proteins, cholesterol esters or cholesterol esters, potentially proteins. We know there are definitely going to be proteins and a small but significant amount of triglycerides. We're actually not really going to see triglycerides in the plasma membrane because they are going to be part of storage. So this is not going to help us. Phospholipids, sphingolipids, cholesterol, and protein with some carbohydrates. That sounds okay so far. Lipoproteins, in which triglycerides are a major component. Again, we're not really going to see triglycerides in a plasma membrane. A lipid bilayer, this sounds enticing, formed mainly from cholesterol with protein attached to both sides. It's actually, while there is a lot of cholesterol, it's not mainly cholesterol. And so this is where this word becomes problematic. Yes, there's cholesterol in there and it's in most animal cells, but it's actually mainly phospholipids. I know. So the correct answer here is actually going to be B. Occasionally we see those sphingolipids. Remember that we're looking for something that has the polarity. So this is where this answer also didn't make sense because we need the polarity in order to form the bilayer. If we throw a bunch of cholesterol into liquid, it's most likely not going to form the bilayer. And yeah, proteins potentially could be attached to both sides. We mostly see them extracellularly. So that, Michaela, could also be kind of a hint as to why. But I wouldn't necessarily like knock that out. When we go kind of like later into signal transduction, you'll see that there can be proteins attached to both sides or potentially even like a protein that's kind of going across the membrane, which is called a transmembrane protein. You may have heard that term before. So yeah, it's good to kind of like think about what is there, but I can see how D could be a little bit confusing. That's why it's usually in there. So one more question here. This is kind of like an application question. So I wouldn't probably predict this exact question to be on a on the MCAT, but it's good to think about. So compared to a typical animal cell, the cell membranes of the paw of a polar bear would most likely have an increased concentration of which macromolecule? Unsaturated phospholipids, potassium channel proteins, saturated phospholipids, aquaporin proteins. All right, so it seems like we're set between A and C. Monica has got, I think, the best kind of clue. It's cold. What do we know happens at cold temperature to fluidity? Cold, it means a decrease in fluidity just based on the temperature. So, right, we're always trying to move towards normal, which means that we want to either have more fluidity, more maybe cholesterol in this case, but cholesterol isn't one of our options. So when we're thinking about, these two are just basically distraction answers. When we're thinking about saturated versus unsaturated phospholipids, something that's going to give us more flexibility and more fluidity is going to be A, unsaturated. So you can see how like it's one of those kind of like multi-steps. Yeah, and you might think about the paw. I think about like the paw pad. If you have a dog or you have a cat, when you flip over their paw pads, even though they're kind of like really rough in surface, they're really soft. And that's because just like our fingertips, that's their tactile movement. So they need that for tactile sensation. So that's why we want to like maintain that fluidity. We also typically, even if you want to like drop the function part, when we're thinking about fluidity, we don't want to err to like an extreme. So if we already know that at cold we have decreased fluidity, we don't want to add anything else in that's going to take it to a next level extreme. We'd rather kind of move a little bit more back towards like that medium normal, if you think about it as like a spectrum from like, fluidity kind of on this spectrum from most to least. So if we know that it's kind of cold in temperature and we're down here, we don't necessarily want to become more extreme. We're trying to move kind of closer to this normal. So what can we add to move it closer to there would be unsaturated phospholipids, kind of add a little bit more space in that membrane and go from there. We're kind of, how are we feeling like time wise? We're about halfway through slides, which is perfect. Do we want to take like a 2-minute brain break? Would you rather keep powering through? What kind of feels more enticing? Okay, we've got 2 goes. I'm going to go with the goes. I'm okay to like keep talking. You might have been able to tell that I'm a pretty like chatty person naturally. Okay, we've got 4 goods. If you need a brain break, feel free to like message me privately. If enough people say, we can always take one in like a couple minutes. All right. So when we think about these membrane transport proteins, we just saw in that question that proteins are part of it. And we talked about we have facilitated and simple diffusion and active. Anytime we're either in facilitated or active, we're most likely using a transport protein. So I'm just going to add that to my slide here. So we're thinking about facilitated, a helper. It makes sense. It's right there, or active. But it's important to think about to yourself. Sometimes we can kind of like jump into these buckets of, oh, if there's a membrane transport protein, it must use energy. That is not true. If it's just a membrane transport protein, all we know is that a protein is helping. That's what we can say about that. It's either going to fall into facilitated, no energy used, going with concentration gradient, or active, going against concentration gradient, using energy. So it's important to kind of just remember that that's not always like a black and white rule. So we have channels, which if you think about a channel, like in a harbor or something like that, that's something that it can just pass right through. So if this was my bilayer, I'm just going to kind of like extend it out over here. And I had, that got rid of the whole thing. So I'll have to redraw it. I might have just, you often see them like this, a little channel where things can just pass on through. And remember that we're always going to go from areas of high concentration to areas of low concentration. That's just the way that science works. I did not make those rules. We all just have to agree to it. So we're going from high to low and we're just flowing through our channel. We're happy as can be. We're going to see another part later where channels come into play. So it's just kind of remember that they're there. And that's going to be technically facilitated because that's a protein involved. But it is, just a protein lined pore and it's passing on through. Compared to if you have a transporter, which often these are enzymes, we know that like the basic rule of enzymes is if something binds, there's a conformational change. So think about these. You may have seen like videos in biology class. Something is binding kind of on this side, which causes this conformational change. And now it's going to be spit out on the other side. And when we think about that kind of going in one direction across a membrane or one solute, that's going to be considered a uniporter. It's just one solute coming in, change, and getting kicked out the other side. Compared to something that is going to be coupled transporters, or often sometimes they're called co-transporters. Those are going to be transporting more than one, typically 2, I would say in most examples, 2 different so I'm going to write that here, co-transporter. Typically 2 molecules. And then you have these terms here, symport or antiport, which are basically, as they sound like, they're going to talk about port is movement and sim, meaning the same direction of movement, compared to antiport, moving opposite directions of movement. That's all that's going to be. None of those, right, are all using energy or all not using energy. These are just kind of examples, depending on the example that's given or the named transporter that's given, you'll know more about whether or not it's going to be using energy or not. However, typically when we see the word pumps, that is a time when you can think that energy is being used. So when we talked about, like at the beginning, I talked about that ATPase pump. This is the example of it here. Basically, we're using the energy from the ATP to actually send through, in this case, it's a hydrogen ion, but it could be something else as well. It could be a really large molecule. It could be a whole protein. You never know. But typically pumps, we can think of energy. We're using energy always. These other examples of transporters or channels are typically not using energy, but transporters in general, that term, could or could not be using energy. So it's just kind of important to keep that in mind. This is another example potentially of what could be considered a transporter, but it is in this case using energy here in the form of light to move this orange molecule from one side to the other. Typically, if we're using energy, we are thinking about going against the chemical gradient. It's a like more than likely chance, but it's not 100%. So it's just important to kind of keep that in the back of your mind. A really good example is glucose. Glucose is a really large molecule. Typically, there's more glucose in the blood than are in cells. We obviously need to get it into cells. So therefore, we're moving with the gradient. We're going from high areas in the blood, from like the glucose that's coming from the food we're eating, and moving it into cells. It's still a large molecule, so it costs energy to actually move it into the cell, which is like part of that cellular, kind of the breakdown of cellular respiration. The first part is moving into the cell, and it already costs energy. So when we think about also other movements, so these were examples of like membrane transport proteins. So the solute is moving via, or the gas or whatever is moving via this protein, this molecule. Compared to, let's have an example where we don't have any proteins, or something in the membrane is happening where we can't actually move the solute. And so our other option is osmosis, which is the movement of water. And so there's typically like three settings for this, or three kind of examples. Maybe we'll start with this a little smaller. So when we're in this state of an isotonic state, right? And remember our water is moving. So if these are my molecules, right? In an isotonic state, water is happy, the molecules are happy. Typically those are in balance. So I drew 5 inside, 5 outside. When we have a hypertonic state, it typically means that while we might have 5 inside, and remember we can't move any of the solutes, outside, we've got a lot going on. And so just very simply, in order to kind of compensate for that, the water is going to move out of the cell, in this case, most likely the cytoplasm. And what we are going to be left with is a much smaller kind of dehydrated cell. It's often shown as like this wrinkly little being. And then that's just like, think of like a prune or a raisin or anything that's been dried in the sun. The water has been taken out of it. That's what it's going to leave. It's not going to be able to be that nice kind of like taut bubble. That's going to be a hypertonic. So we talk about kind of like the water moving out, and that's going to be a hypertonic solution compared to hypotonic, which, as you could probably predict, is going to be the opposite. So you still have 1, 2, 3, 4, 5 inside, but this time we maybe only have 2 molecules outside. Obviously the molecule number doesn't change. It's just the concentration, or the molecule number doesn't matter. It's the concentration. So a lower concentration outside, which means that water from the surroundings is going to move into the cell to create kind of this balance. And therefore we're going to have what's always blown up as like a really large cell, and there is a potential to rupture here. When we think about kind of like either a transport protein or the process of osmosis, so either the solutes moving or water moving, we can see that the theme is common typically, is the goal is to have a more even electrochemical gradient. We're moving from high to low. We're trying to achieve this balance for the most part in either scenario. We obviously are going to, ruin parts of this gradient by doing active transport and using energy to pull something against its concentration gradient. And maybe that's where osmosis kind of comes into play additionally. And that water also, takes on a little bit more fluid to kind of like keep that internal balance correct. But we can see in these two different models, the goal is always to kind of like achieve this nice balance of like peace and harmony and happiness. And that's like something that's kind of like our body is going for peace and harmony. Physics is going for disorder. So you can just always remember physics is chaos. That's my kind of thing. Whereas the body, much, much more peaceful, much more harmonious. That's where I want to be. Most of you, probably as aspiring physicians, want to be in the harmony. So that's what we're always thinking about. So when we kind of think of like some really important proteins that are going to be game players, we talked about the sodium potassium pumps. We're obviously going to see more of these on cell membranes of nerve cells than potentially what we're going to see on like a bone cell or a kidney cell. So it's important to remember that, right, the structure of my plasma membrane is going to reflect the function of my cell. So if I have if they're describing a cell to you, it has a lot of sodium potassium channels. It has a lot of vesicles. It has a lot of projections, and it's not perfectly circular. You're already starting to think neuron, right? That's sounding exactly like a neuron compared to maybe they're talking about a cell that's very flat and very thin. It maybe has like a wax part of it, or a lot of cholesterol as part of it. And potentially has the ability to make a cuticle. We're thinking about something in skin related potentially. So we're going to just keep in mind always that the structure of our plasma membrane is going to represent the function of what that cell's job is and what the goal is for that cell to do. Another thing that can be useful on the outside of cells are glycoproteins. And so these are going to be like basically sugary proteins that are just help identify the cell. Sometimes, like I mentioned earlier in the blood typing example, that they're either you're positive for a rhesus factor or negative for a rhesus factor. I don't know if in repro you mentioned this, but sometimes this can even happen, or this can be like a big enough deal for two people that are trying to have a baby. Potentially, if somebody is negative blood type and the other person is positive blood type, if the embryo that's formed has a positive blood type and starts to show that rhesus factor, like the woman's uterus can actually identify that as foreign, and potentially, it doesn't always happen. Obviously, people are born with different blood types to their moms, but potentially have what's considered like a rhesus factor miscarriage, where actually that like the uterus saw that basically as an invader and becomes very hostile and forces a miscarriage to happen because they can sense that there's like enemy cells in their area. So sometimes also like these glycoproteins can help determine self from non-self. If there's like ever infections, or this is why it's so important with like blood transfusions or even organ donations. Oftentimes they want to match you based on your blood type because they know that like your blood basically just like knows. And if it decides that this kidney that got donated is not, its pal, then adios, it's not going to happen. So it's just really important to remember that those glycoproteins are really powerful in identification. Last one is going to be membrane receptors. And these are just kind of like different things on the membrane that are either receiving messages from the outside or like the other environments, or potentially they're connecting to other cells. We're going to see a few examples later of like membrane receptors that are actually helping connect cells together. And so those are kind of like the most notable ones that we've got. They can also sometimes help with like messaging, or how we like actually get things out onto the cell. So when we talk about exocytosis, we're sending something out. And so what can happen is that you have your cell here, you've made something, you've packaged it up all at the Golgi, and it's heading for the outside. And so as it moves kind of closer to the cell membrane, eventually it will fuse with the cell membrane, and then the contents will leave. So if I put like a little orange in here, and now my orange can leave. That's considered like exocytosis, sending something out. Maybe it's going up to the surface of that cell membrane, but potentially it's actually just being released completely. It's a product that's maybe a hormone, and it's going to be released into the bloodstream and go somewhere else. Or maybe it is a membrane receptor protein, and it's just going to go like stand. If you've ever seen antibodies, they're often like drawn as these little Ys. If you've taken immunology, you know, like all about like showing different parts of different like decomposed, basically like almost what's like trash on your cell. And that can say like, I've either been invaded by this or haven't been invaded by this, or I'm trying to basically produce this kind of immune response by putting something up on my membrane. Alap, is that how I say your name, or Lap? Can you explain what you mean by your question? Are you talking about in any kind of cell or specific cells? Are you talking about like muscles specifically? I'll wait for you to chat me back before I answer that. And I'll go over endocytosis. As it probably sounds like, endocytosis, right? Something's going in. So you're basically like little vesicles can kind of hang out outside of the cell. You might have like a little receptor here. My little vesicle is going to come and land here. It's going to have, you know, once again, when we have things interacting, we know that there's going to potentially be conformational change. This is going to conformationally change. Maybe it's going to bring it down slowly. Then I go back to my other color. Maybe this little receptor will soon be like this, and then eventually it's going to fuse with the membrane and then go in to wherever it's supposed to go. So you're asking about like nerve and muscle cells. Yeah, so calcium channels are just going to be the same thing. Oftentimes we have calcium stores. We're going to talk about something later, the sarcoplasmic reticulum that stores a lot of calcium. So you can have like internal stores of calcium, which make it really high in the cell. So then you want to release calcium from the cell. Like in muscle contractions, you need calcium available in order to do your little muscle stroke. And you also need calcium available in really high quantities in nerve cells, because calcium is also like a stimulant to release the vesicles. So those are definitely also other channels. Oh, that's the word channel, not calcium. Other channels that could be present. There's not really much more in detail that you need to know. You don't need to like know about their function. I would just know that they're present and that you're probably going to have more calcium channels in a nerve cell compared to like a bone cell. Well, there might be a lot in bone cells too. More calcium channels in a nerve cell compared to a skin cell. Can you talk about how exocytosis and endocytosis is different from retrograde and intergrade? Yeah, typically endocytosis and exocytosis happen with vesicles. So they're happening like with something that has a plasma membrane. It's coming into the plasma membrane and then is being enveloped into the plasma membrane. Whereas transport, it might just be like moved across the membrane, if that makes more sense. I can try to explain it again if that does not make sense, Stephanie. So I'd think about it maybe like this. Let me try again. So when we're thinking about endocytosis and exocytosis, we're typically thinking about things that are membrane bound. Whatever's in there, right? Doesn't really matter. Maybe it's liquid, maybe it's waste. It's something is going to be membrane bound. So it's always going to come and meet the membrane and be a part of the membrane to either empty its contents in to the cell or take the contents out of the cell. When we think about transport, oftentimes what is either going up is going to be like a protein going to the surface. So that was probably where I maybe confused you in like that kind of transport. We're having a protein go up. Whereas like I would think about endocytosis, or maybe you've heard about pinocytosis, where like it's literally like the cell is drinking. It's taking cytoplasm and it's just dumping that right into the cell. So we typically like those are going to be things that are like non-toxic for the cell, like maybe something that the cell already has inside, or sometimes like transport is something that like is toxic to the cell. So we don't want to even let that be part of the cell membrane for a moment. We're trying to get it like through, and maybe it's going to be transported across a protein compared to being a part of the membrane and then being outside of the membrane. I think that's probably like where the difference is. Yeah, I would say that's a good way to think about it. So like the endo or exocytosis is like a dumping in or out of the cell, but particularly that vesicle is becoming a part of the plasma membrane and then going out. So it can also like that's how our plasma membrane is fluid. We're adding to it when we're sending something in or out of the cell compared to transport is probably going to be transported through a protein or a channel. It's not necessarily going to morph with that cell membrane, if that makes sense. But yeah, you could also think about it being more centralized and like more simple. Yeah, great question. All right. So little review question for us. I hope these like review questions are helpful just to kind of like break up your thinking. Which statement represents a notable difference between simple diffusion and facilitated diffusion? Unlike simple diffusion, facilitated diffusion can transport ligands, which is just like basically solutes, against a concentration gradient. Unlike simple diffusion, facilitated diffusion can occur in any type of cell. Unlike simple diffusion, facilitated diffusion requires energy in the form of ATP. Unlike simple diffusion, the rate of facilitated diffusion is limited by the number of transport membranes in the protein. A, B, C, or D. Great. D. Really simply, facilitated diffusion, it's still diffusion, so it is not going against. Facilitated diffusion can occur in any type of cell, so can simple. Facilitated diffusion requires energy. That's not true. And that leaves us with D. Excellent. We'll just put an X through there. All right. So we've talked about kind of like our plasma membrane. We've taken a long time to like understand all the components of it. And now we're going to look at kind of then like what is actually inside. And really what's most unique, and this is something that I feel like does come up often, is that a big difference between eukaryotic and prokaryotic cells is that eukaryotic cells have membrane-bound organelles. I feel like it's just kind of one of those like MCAT truths. You've probably heard of it. So we're just going to kind of go over the membrane-bound organelles, remembering that if they have a membrane, they're also going to be affected by fluidity, be affected by temperature. We're thinking about why they have a membrane and kind of all of these different things. And potentially then we're going to look at some other support players of the membrane. So the cytoskeleton, is what's considered like a support player of the plasma membrane. So it's both for like the skeleton, like we think about like what our skeleton does for our body, it's providing us structure and support. It's why we're upright and not just like a blob. And the cytoskeleton does the same for the cell. And there are going to be different parts of the cytoskeleton. So we've got something called a microfilament, and these are made of actin. My way of remembering this is that microfilament, let's use the highlighter, microfilament here is made of actin. And this, I feel like you have to know, it's just something that pops up all the time, particularly in multiple choice. So it's just good to like always know that. And actin is going to act in cell cleavage. So it's all about like kind of binding together, pinching things off. And it's going to be really helpful in that. Compared to microtubules, which are made of alpha and beta tubulin. So those two kind of like pair well together. And they kind of form like, they definitely have a supporting role in kind of like shape and structure of the cell, but they also, the microtubules are considered like this transport highway is always how I like to think about them. Or like a tightrope, you may have heard them being called. And so you have the kinesins, which are going to walk towards the cell periphery, which is considered like the positive end, and the dyneins, which walk towards the cell center. Someone told me, I think it was Justin who taught my bio section, that if you think about like the center of the cell as the drama, that's like the negative space. So the dyneins are walking towards the center, which is like the negative pole. Intermediate filaments are also part of the cytoskeleton. They're going to kind of like support things and they're going to help make connections. We'll see that when we look at cell connections a little bit later. And then we have these kind of like movement, I would say like movement additions. And they technically are made of microtubules, which is why they're kind of included in here. We have cilia and flagella, which both follow this 9+2 organization. I remember first hearing about this in AP biology and just thinking like, oh my gosh, is this like a load of garbage or what? But it's kind of cool at the same time. But it's just one of those things you have to remember that 9 + 2 are cilia and flagella. And so you can see here, there are 9 pairs on the outside, plus the 2 in the middle, which is why it gets its name, 9 + 2 microtubule organization. Compared to here, these 9 triplets, and this is in centrioles, which are going to follow a different kind of pattern of organization. And it kind of, I don't really think this structure in a way helps you, like if you saw the 9 + 2 to know, oh yeah, cilia and flagella are more involved in movement, but the way that the actual microtubules kind of grow out from that does make a little bit of sense. And so when we think about cilia, typically those are lining either like the surroundings of a cell. So often you see cilia as represented as like little short hairs on cells. So sometimes they brush things away. Your respiratory tract is lined with cilia. The fallopian tubes are lined with cilia. Sometimes in your digestive tract, there are also cilia, and they either like brush different, the fluid along, or they're in your trachea. They're really important for like moving dust out, or potentially even moving liquid, when you swallow. If you've ever swallowed like a pill and not had a lot of water and it kind of feels like it's stuck in your throat, that's actually your cilia being disrupted in your trachea. And so even when you like feel like you swallowed it fully, or sorry, not in your trachea, in your esophagus. But even when you feel like you've swallowed it fully, it still feels like it's sticky there or like something's there. And that's like in your esophagus. It's also there as well to kind of like help move things either up or down. Sometimes when you're coughing, you can feel it kind of like coming up a little bit, and that's it getting stuck in those cilia. So just kind of an interesting application of them compared to flagella. The famous application is sperm. Often flagella are like we see them in bacteria, and they are going to help like move around an environment. These like whipping tails is always what they kind of describe it as. So obviously our little sperm. And then centrioles and microtubule organizing center. I always thought this was like kind of confusing when I first like would hear about it, but typically it's like a zone near the nucleus, and really it comes into play when we're having cell division. So I guess it's not only mitosis, but also meiosis. So really it's more like cell division is when it comes into play. And the centrioles in this like center, they are going to replicate. And then as we've, replicated all of our DNA and we're kind of getting ready to kind of move to the different poles of the cell, the centrioles are actually what's going to be to send out the mitotic spindles. So if you remember like lining or seeing all those pictures of, all of the chromosomes lined up, and then having these like, they're usually like drawn as like little X's or two little circles or like little barrels next to each other. And they have their branches. And those are going to be the mitotic spindles that are then going to shorten one by one. This is where centrioles come into play. Those are also going to be part of this cytoskeleton cell movement kind of bundle. I would say it's really good to be really comfortable recognizing these terms, kind of understanding simply like what do they do, what's their role, and then for like the cilia, the flagella, and the microtubule centrioles, understanding either that they're 9 + 2 or the 9 triplets. Sometimes that just comes up, and like that filaments are actin, tubules are alpha and beta tubulin. This is kind of a little picture of our microtubule. And you can see the kinesins towards the cell periphery. So we consider this the positive end, and dyneins towards the center. You can always think of the center as the drama, and dyneins are drama. So that's going to be the negative end, if that helps anyone. So that's kind of like what we're thinking about when we're thinking about cell movement. Another place that potentially some of these cytoskeletons, particularly the intermediate filaments, come into play are going to be with cell connections. And there are a few different types. Oh, sorry. What does that say when you say positive and negative? Oh, just it means towards the center of the cell. It's just how we kind of, this also looks like an X, towards the center of the cell compared to towards the periphery of a cell. Kind of like in a cell, you don't have north, south, east, west. You have positive and negative. So it's just directionally. Nope, doesn't have anything to do with DNA. It's just kind of like directionally in the cell. If they say, oh, it was moving in the positive direction in a question, you just know like, oh, it's moving to the outside of the cell. Because maybe that's it. Maybe the question will be like, a kinesin is moving in the positive direction in a cell. What's the most likely, like event that's going to happen next? Endocytosis or exocytosis? Well, we know if it's moving towards the periphery, it's probably taking something out rather than something coming in. That's like a really poor question, but kind of something like that. So it's just directionally to like help set the scene. So when we think about these different kind of like different connections or different filaments that are going to be coming into play. We talked about what they're doing when it's just one cell, but we can also have cells that communicate with each other, cells that live near each other, cells that are connected. This is definitely important in our physiological tissues. And so obviously we have to have interactions between these cells. So we can have something called a tight junction, which basically is a full sealed like connection between two cells, but nothing passes through there. Just think of it as kind of like an anchor. These two things are connected together, compared to adherins, which are using actin. It's another nice AA, adherins and actin. And they're also going to anchor, but there is, you can see kind of in the picture here, there's a gap still between those cells. Then you have desmosomes, which are going to connect via intermediate filaments. So it's those intermediate filaments coming back into play. Gap junctions, which sound exactly like they are. They're a gap between the two cells. Think of this like a channel. And this is really important because if there's a channel, then basically the like cytoplasm of those two cells are connected. They can send things to the other cell. And so sometimes we see this and particularly, I think in plant cells, they can kind of like share their fluid. They can obviously share like anything that can freely pass through there, typically small, seeing as like no one really wants to have like a huge channel in between. They'd probably just merge and become two, like one big cell then, but have to get rid of a lot of contents, obviously. And then there's something called a hemidesmosome. These are probably the least common. And that's where the intermediate filaments are anchoring it to the extracellular matrix, which is just kind of like other tissue around. And these kind of cell connections are just important to like think about. They do sometimes come up, but they're not, super, super common. They are important when we think about like epithelial cells. Epithelial cells are one of the 4 tissue types, and they are often making linings in the body. And so if we have a lining, think about your skin, think about like your respiratory tract, your digestive tract. We want all of those cells to be really well connected together. So we're most likely, well, here I'll ask it as this question. With epithelial cells, we want all of those cells to be really tightly connected to each other. What are we most likely going to use? A tight junction, a desmosome, or a gap junction? Yeah, we're going to use tight. We want those to be absolutely tethered together. The benefit is that then they stick together. The downside is if you damage one, potentially if there's tension on one, it's tension on all of them, which is like why wrinkles happen. So you're kind of pulling them all in one direction or the other. But tight junctions are going to be really helpful in that. If we have like cells that need to communicate with each other all of the time, gap junctions are really common in plants because they are, sending stuff back and forth to one another, sharing nutrients, sharing water. Potentially this part of the leaf, is more exposed to sunlight than this part of the leaf, so they can like share between them and between those cells. So gap junctions, you definitely see a lot in plants, but they are still in animal cells as well. So we're gonna keep going on here. So yeah, we talked about like that they're protection, they're anchoring, but they also can help with kind of like keeping those channels open for communication. So we're always thinking about how does this structure impact the function, or why am I kind of using this? So then when we kind of like move into the membrane bound organelles, a lot of this should be review. So I don't want to necessarily like speed through it, but I may not like spend as much time kind of going deep because there is a little bit of new information at the end, or kind of like maybe less commonly covered information. So when we talk about this endomembrane system, it just means membranes that are inside. So we're thinking of those membrane bound organelles that are going to be inside the cell. So we're thinking about our nucleus, which has a double membrane, our endoplasmic reticulum, and our Golgi. And the three of these kind of work together. They're like in a little zone, usually all together. And that's because some of their functions are very closely linked. And basically, this is like a big, like, it's often like the Golgi is like the post office, and the endomem or the ER is kind of like a factory. And so there's a lot of things being made, a lot of things being packaged. We got a lot of stuff going on. We remember that inside the nucleus, no protein synthesis can go on there. So if there are proteins that end up in the nucleus, they've been imported from somewhere else. So that's just like an important thing to remember when you're thinking about what's going on, because sometimes they'll say like, oh yeah, there were like some ribosomes inside the nucleus. Well, if they're there, they've come from somewhere else. And so the nucleolus is kind of this zone of ribosome synthesis. And so anything that's being part of ribosome synthesis has been imported after being made outside, which does seem like contradictory because it always has to get made, chicken and the egg, but you just have to go with it. So the nucleolus, right, is our site of ribosome synthesis. We're going to have a ton of ribosomal RNA that's going to be producing these ribosomes. They have very specific genes that ribosomal RNA is coming from. And then they're going to be imported into the nucleus, or so that they can kind of be in that same region. And if we have a cell that's undergoing a lot of protein synthesis, we would expect that nucleolus, or that area kind of around the nucleus and where we're having our ribosomes made, to be really active and really quite large. Then we're kind of going to go to the endoplasmic reticulum. You've probably heard there's smooth and there's rough. Rough has ribosomes, smooth doesn't have ribosomes. We know that ribosomes function in translation and protein synthesis. So we're always thinking about why are there ribosomes on the RER? Well, it's involved with protein modifications and making proteins and folding proteins. So that's important that there's ribosomes there. And those are typically destined for the plasma membrane or excretion, compared to the smooth ER, right? No ribosomes. So we're not talking about proteins when we're in the smooth ER. And that makes sense because it's got different functions. It's going to be processing or forming lipids. It's a big site of detoxification and processing like drugs and detoxification of drugs specifically. So there's a lot, we see like larger smooth ER in liver cells. There's also the conversion of G6P to glucose during gluconeogenesis is happening here. And that's because the G6P is actually in the membrane. So it's like an endomembrane protein in the smooth ER. And then we also got calcium storage here. Think about like the sarcoplasmic reticulum. So maybe when we have those calcium channels open in the cell out here, there's going to be a vesicle that then is also going to have a calcium channel. That vesicle then is going to end up merging with the endoplasmic reticulum and potentially storing some of the calcium there. So the smooth ER is doing a lot more than the rough ER. I would just know like a few of these off the top of your head so that you can kind of think about when you hear about smooth ER, or you're thinking about a question about comparing the difference between smooth and rough, you already have some ideas that are coming to mind, and you're not just like completely lost as to what's going on. So once we've kind of got our packages ready, or potentially our proteins ready, they're going to go to the Golgi, which you can see, oh, sorry, I drew this actually going to the wrong place. Let me not confuse you. So potentially there's calcium channel here. It's going to be stored here. This is where calcium is going to be stored. This is smooth ER on the outside here with no ribosomes. Rough has these little polka dots, RER, compared to smooth. And you see those RER or SER. This is probably a review. Golgi is kind of like our post office. It's accepting packages, it's delivering packages, it's kind of organizing, potentially repackaging. And so I always like to think of this as like the post office, which does seem simple, but I think it really works because you also have to remember that you've got your cis face and your trans face. We see these words a lot in biology, but the cis face is basically going to be where things enter, potentially from the endoplasmic reticulum, and potentially then maybe are processed through the Golgi and then are going to be sent to the transface, which is like our exit face, which is usually closer to the plasma membrane itself. So, you can think about this as like, if this was a post office, right? The endoplasmic reticulum is going to be facing, that's like on the street side. I enter there, I have my package, I give it to the post office workers. They've labeled it, they've packaged it, maybe they've repackaged it. It then goes behind these mysterious doors. I'm sure we'd all love to know what's behind there. It gets sorted and resorted and eventually put on a truck to leave out the back and go to where it's going. And that's going to be the transface, is that back kind of side of the Golgi. And so typically what's going out here are things that are maybe going to be a lysosome. So maybe it's like a new vesicle that's going to be taking in different protons and acids to form a really acidic environment. Maybe it's something that's going up to the plasma membrane, so it needs to be sent there. Maybe it's a vesicle that is destined for another location. So those are kind of things leaving. We talked a little bit about ribosomes, and this may have been covered in another lecture, so I'm not really going to focus on it intensely. But ribosomes have two units. There's the large subunit and the small subunit. It's really important, I would say, to know these sedimentation coefficients. They seem to pop up a lot, but basically, if you took a bunch of ribosomes and put them put the cell contents in a liquid and centrifuged it for a really long time, you're going to get, there's obviously a lot of ribosomes in a cell, but you're going to get different bands. And you can tell basically if you have a lot at 80S, you know that you have a bunch of intact ribosomes compared to if you have things at 60S and 40S, which would mean you have like ribosomes that you have the parts of. And then you can also tell the difference between eukaryotic and prokaryotic cells depending on their this band weight of their ribosomes. So I would just know these numbers, both the 60 plus 40. Oh, that is not, maybe this is, hold on. Let me check this really quick because 60 plus 40 is not 80. So I want to make sure I give you the right information. Oh, it's weird. It's not like math. Okay, I remember this now. Even though the big is 60 and the small is 40, together they are 80. It's kind of like when they interact, they almost in a way like lose part of their quote unquote weight. This is not their weight, but it's just a way that it's like where they fall in the sedimentation lines. So don't think about it as weight. Think about it just as like where they fall, if they're either just small, large, or the whole. And typically, yeah, the small subunit is like on the bottom. So you should remember 60-40 as the large and small parts. Sorry, did that disconnect again? So don't think about it. Sorry, I kind of confused you. I confused myself as well. It's not necessarily like math. It's not like weight. Just remember, if I have a bunch of 60S, like when I do my sedimentation, I have a bunch of things coming up at 60S, that's going to be the large subunit, and that's a eukaryotic large subunit. If I have a bunch of molecules showing up at 30S, that is going to be my small subunit of a prokaryotic cell. So sometimes they will show you, is this something you guys have gone over before? I could kind of explain it like a little bit more slowly. I just thought this was somewhere in a different lecture. Does this seem vaguely familiar, completely foreign? SOS. I'm presuming that's not new. Remembering it? Okay, so I'll kind of keep going. But potentially, I would say the way that you would see this in a question is you'll see they'll either give you like 4 vials, and they'll say, these are contents of cells that have been centrifuged. Use the sedimentation coefficients to like answer the following questions. So you might see, they'll be like this one, this band, and then these two. And they might say like, which of the following containers contains like parts of ribosomal subunits? So you might say, well, these two basically are if this was A, you might say A and B show about the same, and they are typically like higher up in the container is a higher number, and they're usually labeled. So we'll say, well, like, well, these guys both contain 80, so we know those are whole subunits. So those are not going to be, you know, parts of the subunits. Compared to when we see two bands, we know that then we have two different subunits. So these are going to be potentially large and small in this one labeled D compared to C, which just shows also only one band. And we know that the different subunits have different sedimentation coefficients. So they need to be at different parts of the vial. So this is how you can kind of use these. So I would remember that you need 60S for the large subunit and 40S for the small in a eukaryotic, and together they come up at the 80S sedimentation coefficient. And for prokaryotic, 50 and 30 for large and small, and 70 for the whole. You're just going to come over these. It's going to be one of those things. If you have it already remembered, it's going to make you so much more confident. If you don't remember, it's a lot harder to like pull things together or figure out what's going on. So these guys are definitely important. We obviously know that they're in polypeptide translation. And it is important to remember that if the polypeptide at the ribosome that's currently like going through and being produced has what's called a signal sequence, it's basically going to be helped by this little chaperone called a signal recognition particle, an SRP, and it will move into the ER to finish translation, and most likely the rough ER, because remember, we still need a ribosome to finish translation. So we're probably going to go to the rough ER. And then it's good just to like think about the fact that like there are free floating ribosomes that are just hanging around doing whatever they need to do. But there are also bound ribosomes like we find on the rough ER. And so while they both are involved in peptide translation, potentially there could be differences in what types of proteins they are producing, or potentially like this is doing kind of like in this translational mod, like almost like post-translational modifications or mid-translational modifications that are going on. So those are just kind of like some important things to remember about ribosomes. Another membrane-bound organelle, obviously the mitochondria, powerhouse of the cell, everyone's favorite organelle, I hope, you know, it does it all. Kind of the big things to remember are that there is mitochondrial DNA. And this is if you've taken any of your 23andMe, they will tell you like your maternal haplotype. And that's from your mitochondrial DNA. There's like also this idea that we all eventually came from this one mitochondria DNA called mitochondrial Eve. And so remember that we do have that in there. And it does code for some proteins on the electron transport chain. So that in order to get from DNA to protein, we're going to need ribosomes in between to help us out. So there are some mitochondrial ribosomes. However, 99% of the proteins that are functioning within the mitochondria are like encoded for by genes in the nucleus of the cell. So they're imported into the mitochondria. But if you think about like some of those ones in the electron transport chain that are hanging out, those were made in the mitochondria. Those are from the mitochondrial DNA. They have not been imported into the mitochondria. So, and remember that, you know, it's this double membrane again. We've got our outer membrane and our inner membrane. And that comes into play like when you talk about kind of where different steps of cellular respiration are going on. Are they in the cytosol? Are they in the matrix? Are they in the membrane? You know, all of those kind of different things. Are they in the intermembrane space? Are they crossing the membrane? Those are some good things just to be kind of like aware of. Right. Next, other membrane bound kind of things are these lysosomes, which we've kind of touched on a few times. Basically, think of them as like little stomachs within your cell. They are going to be super acidic, just like our stomach. Their goal is to break down different things. Sometimes it's waste. Sometimes they're breaking something from a bigger kind of molecule into smaller molecules. Sometimes they are being formed around poisons and are going to be able to be released. Potentially, if a lysosome was ruptured, that increase in acidity in the inside of a cell would actually cause that cell to undergo what's considered autolysis, auto, meaning it's automatically happening. Lysis is like another word for breaking. So basically it would like auto break. And then apoptosis, which is basically like the cell deciding to pop, which doesn't truly just like pop. It's very programmed and organized. But basically that means like the cell is going to break down and that cell is no longer going to keep on living. It's going to like actually die. So this is part of like programmatic cell death, which we'll cover hopefully at the end here. And then we've got peroxisomes, which they are specifically breaking down reactive oxygen species. So think about like peroxide is a reactive oxygen species. So it's basically taking peroxide, which we might see as 2H2O2, and then it's going to convert it into something that's much less harmful for the cell. It's going to be water and oxygen. So lysosomes and peroxisomes, both really helpful when they're doing what they're supposed to be doing and helping us out. But obviously, if they leak their contents or they decide to kind of like turn against the cell, there can be some pretty serious consequences that are going to come from that. I've got 2 questions after this. If it's okay, we're just going to skip on because I want to make sure I get you out of here right on time. So we're going to skip over those. If we have time after, we can come back to those. So we're going to skip ahead to the next section, which is called biosignaling. So we've talked about kind of the plasma membrane, what's up on the plasma membrane. We've now talked about the different membrane-bound organelles within the cell and how they're all doing different jobs. But obviously we know that cells are not, or at least our human cells, animal cells, are not living like on their own as just one single cell, not interacting. They're definitely interacting with other cells in their environment. And so biosignaling becomes really important. So both internally, we have something called oncogenes within our cells that are genes that have the potential to turn something into a tumor. Which basically is a tumor cell, which is a cell that is going to multiply and divide incessantly. And so sometimes sometimes these changes in DNA basically will signal the cell that it's no longer self and it's not recognizing itself. And some of like the auto regulation programs are not going to go. And so then it will kind of turn back on itself and induct apoptosis and lyse itself and like go through programmatic cell death. However, obviously, sometimes these genes are a little bit more sneaky. And so therefore, when they are like overexpressed, they can just kind of keep being expressed, get by all of these different checks and balances. And then that grows into a tumor, which could or could not be cancerous. And then the idea that potentially one of these cells breaks off and travels in the bloodstream to somewhere else in the body and then infects there is how we like determine forms of like metastases. And if the cancer has metastasized is like a big word. So these oncogenes, a lot of them have to do with like cell kind of like replication and checks and balances in there. And so when they get kind of like overexpressed, then that can lead to like, honestly, it leads to both negative paths, but one is probably a lot more outcome wise negative. Tumor cells becoming more and more replicated, which is forming a tumor, forming a mass. Obviously, that cell's not doing what job it was supposed to be doing. So we're eventually going to have some physiological change. Other option, which is technically quote unquote bad, but better in the long term, is that cell then is going to die. And don't think about it as like when we think about cell death, we often think of necrosis, but it's not necrosis. It's very programmed. It's going to do it in as like the easiest way as possible. Because I sent out the PDF of these, it didn't really like share correctly, but there was like a little GIF. But basically, the nucleus condenses. It kind of starts to form these weird shapes, which are called blebs. And the blebs all just kind of become membrane bound, like little parts and pieces of the cell, but it's not all a complete cell. And then neighboring scavenger cells can come and kind of like chomp them down, recycle them. And that's how we keep going. So, as I said, programmatic cell death is like really regulated. It's really safe. It's very like a procedural thing. Obviously, if all of your cells are spontaneously going programmatic cell death, we're going to notice that. But it can either be apoptosis, which is kind of that process I just explained, or autophagy, which is actually where like the cell itself eats itself. And that's kind of an interesting process of like self endocytosing and recycling. compared to necrosis, which is going to be the result of injury or damage to those cells. And that's necrosis and sepsis and things like that you'll see later kind of in a physiological kind of setting. But as I said, we know that cells just don't like hang out by themselves and live by themselves. They're obviously in an environment with other things. So there are different kind of signaling cascades. I would just be familiar with the names of these different cascades and kind of like overall that these are options. I wouldn't spend time like going into like extreme depth of all of these unless you have a year before your MCAT. But just good to know you have hormones. There are definitely like different types of hormone signaling, autocrine, which is basically like you're self-signaling to your cell, paracrine, you're signaling to a neighbor, endocrine, you're releasing something into the bloodstream, or exocrine, you're releasing something into the duct. Think about your salivary glands. They're producing like hormones, or the hormones that are impacting them are going to secrete the saliva. Saliva goes into the duct, not into the bloodstream. And that's like the product that's coming from that signaling. Signal transduction is just a fancy way of making an outside message like an inside message. So something that comes to the outside of the cell, interpreted by the cell, is going to change the intracellular actions or behavior, or maybe the products that are formed. And those are kind of done by some of these key players: G proteins or G protein coupled receptors, GPCRs, cyclic AMP, protein kinases phosphorylate things. That's like a basic principle. And basically that can change the function of a protein. Sometimes phosphorylation increases activity, sometimes it decreases activity, sometimes there's no change. So you don't really need to know the specifics, more just know that these are some of the players in the game. And then GTPases, which similar to any other ACE or like an ATPase, they're just going to hydrolyze GTP, and that can either sometimes turn on or off the G proteins. So these are just some of the like players. Just read over them, be comfortable kind of identifying and knowing that they're part of cell signal cascades. As we said, cells don't grow alone typically in a human. They're kind of in constant communication with like their surroundings and other cells near them. We talked a little bit about stem cells last time, and so those early signals that can be given to those embryonic stem cells potentially are going to influence the future of their lives or what type of cell they become. And that step is called determination, which is a really important word to know. And then after determination, after this cell is told you are going to be X type of cell, we obviously need to modify potentially what's in the cell or on the surface of the cell to make sure that that cell is actually ready to do that job. So that word is called differentiation. So determination, I would say, like, is kind of getting sorted into your Harry Potter house, and differentiation are the classes and the games and all the other stuff that you play like while you're at school to become the person that you're going to be. Sorry if you don't like Harry Potter, but that's about the best I can do. And then, as I kind of mentioned earlier, we have these four, basic tissue types. So when we're talking about determination, potentially it's into a tissue type. Potentially it's more into like a specific cell of like, you're going to be a neuron in this region of the brain with this type of, like different neurotransmitter. So you can kind of like determine those kind of different things. And those four tissue types are epithelial, muscle, connective, and nervous. That's for animal. Just want to make sure. Can I even spell? This is what happens after 2 hours. Animal cells. Oh, is it really? Has it moved on now? Is it frozen? Sorry. All right, let's see if we can reshare this. Of course, right at the end, he wants to just hang out. Let me stop mirroring and start it again. Sorry about that, guys. We might go like 2 minutes over. I'm really sorry. I really wanted to finish right on time. All right. Sorry. I was just blabbing on and you couldn't even see. Thanks for letting me know. So yeah, that's the difference between determination and differentiation. And again, the determination is happening super early on. We don't have like groups of stem cells just hanging around our body. Obviously, in our bone marrow, we have a few, but other organisms like store stem cells in their body. And that can be helpful when we come to something called regeneration. I'll kind of zoom out so you can maybe see both. Regeneration is where basically organisms can replace lost or damaged tissues, and they can do that either completely. Salamanders are usually the go-to example. If they lose their tail, they can grow another one. Humans have the ability to incompletely regenerate our cells. If you have any scars on your body, you know that that scar does not look the same as the tissue that was there before. It's now different. It's more keratinized. It's filled the hole, right? You're not still bleeding. You're not still having kind of a wound there, but it is incomplete. It's not the same. Those are just the difference between those two words, just kind of like vocab stuff. Physiologically, it's important to remember that not all tissue in the human body is regenerative, which is why heart attacks are so serious, because the heart tissue is a very like common example of tissue that doesn't even have incomplete regenerative abilities. If you have a heart attack and that muscle does not get blood in that area of the heart, it's dead. There's no coming back. It's not going to do a different type of muscle cell. It's dead. Compared to like if you resect a liver, parts of cirrhotic liver or fatty livers, sometimes you can take parts of it away and basically the liver will regenerate enough to continue going with life at a certain point, of course, but it's not necessarily going to be exactly the same, but it is going to have functional cells that are helping it kind of move and grow on. Last slide, sorry for being right on time, is just the concept of senescence, which I know this kind of seems like weird and added in, but it's in this topic. And senescence is the shortening of telomeres. So when we have our gene of interest that is going to be at some point on our DNA, basically there are these extra protective caps on the end called telomeres. And every time that gene is copied, because the DNA doesn't perfectly get copied exactly the same way every time, sometimes there's mutations. Obviously, the way that we're kind of reconnecting the DNA, it's going to be shortened. And so typically we have these telomeres so that the next time when we've still got our gene here, nice and happy, maybe our telomeres are going to be shortened a little bit. That this is a dramatized kind of picture. And so eventually over time, as our cells are older and older and we've replicated and replicated, these telomeres are going to get shorter and shorter, and potentially that could impact the gene. And so this contributes to cell aging, which contributes to the aging of our whole kind of like bodies and the ability to continue going. They think that maybe the Alzheimer's potentially, or like not Alzheimer's specifically, but memory loss potentially is a shortening of telomeres as well. There are a lot of they're doing a lot of study right now on telomeres. You've probably, if you're in research, you've heard about this. And so these kind of like telomeres get shortened. There is an enzyme, telomerase, that can prevent the shortening, but a lot of somatic cells don't make telomerase. So it's kind of like a one in one hand. Should we be like taking pills of telomerase? Well, there's probably like confusion there. So it's just good to like be aware of these words. I haven't really seen these like come up super popularly, but it's good to be there. And they could be actually, one thing that's interesting is that telomerase could be really present in cancer cells, which could explain that basically like when a cancer cell kind of like hijacks the cell's machinery to divide and multiply endlessly, that it could be because of the telomerase is able to like prevent the gene from shortening, that's like basically out of control. Could add to it, but it would be really cool to be able to like do like telomere therapy or be able to like prevent the shortening of telomeres, and just kind of like maybe even slow or reduce the detrimental impacts of aging. So that is kind of the end of our lecture there. The one thing I do want you to vote on, if you want to come to the review lectures, do you want it to be which of the following options? I'm also happy to answer any other kind of questions. Option one, if this will show, would be Saturday and Sunday for reviews, compared to option two would be Sunday and Tuesday, compared to option three, which would be like Tuesday or Saturday, Sunday. Ideally 1 hour reviews, one on genetics, one on metabolism. Let me know. I don't know if you have finals. I don't know if you're traveling for the holidays. I don't know if you have zero desire to review at this point and you're just like, Emma, leave me alone. But let me know if you have any strong kind of opinions. We've got one of each one, a second for three. All right. Okay, we've kind of got it all over. So I might try to do, it seems like option one and option two are kind of popular. Um, option three also popular, but I feel like most likely it will be option two or option three based on that. Okay, I'll send it out. Maybe I'll try to make like a doodle poll or something. And that way, like I can also like you can have time to review kind of your schedules and things like that. Any questions from today's lecture? I'm really sorry to have gone over, so I understand if you need to leave, but let me know if you have any questions. Thank you so much for all joining. I really appreciate how like participatory everyone was today. Hopefully pace was better today. Definitely let me know if you have any feedback. I really want to hear from you guys so I can prepare these review lectures. And I'll send an email probably late tonight. Yeah, thank you.